What is the largest bone in the body? What are the different types of joints? What are the four types of movable joints?
Ocean plays an important role in the formation of fossil fuels. Exploration of gas and oil form in the sea over a period of millions of years as the remains of animals and plants sink to the ocean floor. Non-renewable sources makes up 85% of worldwide energy usage from sources that eventually will be depleted. For today's lesson, let's explore non-renewable energy sources, its advantages and disadvantages.
What do you think non-renewable resources are? Non-renewable resources are coal, petroleum,
And natural gas.
A non-renewable resource is a natural resource that cannot be remade or regrown at a scale comparable to its consumption. An example of non-renewable energy is nuclear energy. It is a non-renewable resource because once the uranium is used, it is gone.
Coal. Coal is a black or dark brown combustible rock made primarily of carbon. It was formed millions of years ago when ferns, plants, and trees died and fell into swamps. The swamp conditions prevented the organisms from decaying completely, and after millions of years of intense heat and pressure, coal was formed. Coal is classified into four main types or ranks based on carbon and heat content: lignite, subbituminous, bituminous, anthracite. The general rule is that the higher the grade of coal, the cleaner it burns and the more versatile its uses. Coal is extracted from the earth through underground mining or surface mining. The choice of mining method is largely determined by the geology of the coal deposit and its distance from the surface. Underground mining currently accounts for a larger share of world coal production than surface mining. Coal can be burned for heating or to produce electricity. To convert thermal coal to electricity, it is first milled to a fine powder, which increases the surface area and allows it to burn more quickly. The hot gases and heat energy produced from combustion converts water into steam to run a turbine and generator. High-quality coal is also a useful raw material. For example, it can be converted to coke for steelmaking. Coal can also be converted to liquid or synthetic gas by advanced chemical processes, making it a possible but costly replacement for natural gas or liquid fuels for transportation. Coal is a highly abundant and cheap energy resource. Coal has powered the industrialization of many nations over history and continues to today. It is a big player in today's energy system, providing 40% of the world's electricity. One major concern with coal is the mining practices used to extract the resource. Ecological impacts and human safety issues, both for workers and neighboring communities, are growing concerns for the industry. Coal is the most CO2-intensive fossil fuel when combusted because it is composed largely of carbon. Coal also contains other elements that cause pollution problems, including sulfur, nitrogen, mercury, and heavy metals. SOx is a leading cause of acid rain, and NOx emissions contribute to smog. In addition, particulates from coal combustion can be harmful to human health. Concerns about climate change from greenhouse gas emissions have put a spotlight on coal plants and have prompted the development of clean coal technologies like carbon capture and storage. That's coal. Oil. Oil, otherwise known as petroleum or crude, is a thick black liquid composed primarily of hydrogen and carbon. The physical properties of oil, such as its thickness, vary greatly depending on the specific combination of hydrocarbon molecules. Oil also contains trace elements of sulfur, nitrogen, and oxygen. Today's oil deposits were formed millions of years ago when dead marine organisms sunk to the bottom of the ocean bed and were buried under deposits of sedimentary rock. After subjection to intense heat and pressure, these organisms underwent a transformation process by which they were converted to oil over millions of years. This is why you may have heard oil referred to as a fossil fuel. Oil is found in underground geological formations called reservoirs. The rocks found in a reservoir have various physical properties that allow them to hold hydrocarbon reserves. Through exploration activities such as seismic, surveying, rock core sampling, and other advanced technologies, geologists locate oil reserves. Oil is extracted from the reservoir most commonly by drilling a well. Once recovered, oil is transported by pipeline, ship, rail, or truck to a refinery where it undergoes a complex refining process that creates petroleum products like gasoline, diesel, jet fuel, asphalt, and many more. Currently, the world uses oil primarily to power its transportation system and to create commonly used synthetic products like plastics and petrochemicals. The production and use of oil presents social and environmental challenges. Producing oil causes land disturbance, sometimes in environmentally sensitive areas. Oil-powered transportation systems contribute to global greenhouse gas emission levels. And control of oil resources is a persistent factor in geopolitical tensions globally. That's oil. Natural gas. Natural gas is primarily methane, or CH4, with smaller quantities of other hydrocarbons. It was formed millions of years ago when dead organisms sunk to the bottom of the ocean and were buried under deposits of sedimentary rock. Subject to intense heat and pressure, these organisms underwent a transformation in which they were converted to gas over millions of years. Natural gas is found in underground rocks called reservoirs. The rocks have tiny spaces called pores that allow them to hold water, natural gas, and sometimes oil. The natural gas is trapped underground by impermeable rock called a cap rock and stays there until it is extracted. Natural gas can be categorized as dry or wet. Dry gas is essentially gas that contains mostly methane. Wet gas, on the other hand, contains compounds such as ethane and butane, in addition to methane. These natural gas liquids, or NGLs for short, can be separated and sold individually for various uses, such as in refrigerants and to produce products, like plastics. Conventional natural gas can be extracted through drilling wells. Unconventional forms of natural gas, like shale gas, tight gas, sour gas, and coal bed methane, have specific extraction techniques. Natural gas can also be found in reservoirs with oil and is sometimes extracted alongside oil. This type of natural gas is called associated gas. In the past, associated gas was commonly flared or burned as a waste product, but in most places today it is captured and used. Once extracted, natural gas is sent through small pipelines called gathering lines to processing plants, which separate the various hydrocarbons and fluids from the pure natural gas to produce what is known as pipeline-quality dry natural gas before it can be transported. Processing involves 4 main steps to remove the various impurities: oil and condensate removal, water removal, separation of natural gas liquids, sulfur and carbon dioxide removal. Gas is then transported through pipelines called feeders to distribution centers or is stored in underground reservoirs for later use. In some cases, gas is liquefied for shipping in large tankers across oceans. This type of gas is called liquefied natural gas, or LNG. Natural gas is mostly used for domestic or industrial heating and to generate electricity. It can also be compressed and used to fuel vehicles and as a feedstock for fertilizers, hydrogen fuel cells, and other chemical processes. Natural gas development, especially in the United States, has increased as a result of technological advances in horizontal drilling and hydraulic fracturing. When natural gas is burned, there are fewer greenhouse gas emissions and air pollutants when compared to other fossil fuels. In fact, when used to produce electricity, natural gas emits approximately half the carbon emissions of coal. Despite fewer emissions, natural gas is still a source of CO2. In addition, methane is a potent greenhouse gas itself, having nearly 24 times the impact of CO2. During the extraction and transportation process, natural gas can escape into the atmosphere and contribute to climate change. Natural gas leaks are also dangerous to nearby communities because it is a colorless, odorless, highly toxic, and highly explosive gas. That's natural gas.
If you ask many individuals, they may say that writing a research paper is one challenging and meticulous task, but with enough practice throughout the years in school, it could be much easier once you get used to it. So, for today's lesson, let's discuss how to prepare a research title.
Here are the things we need to consider. First, a research title should reflect the scope of the study.
Limited to 12 to 15 words. Minimize the use of articles. Catchy and interesting. New and crispy. Must have international characters and reflect the most important results and objectives.
What are the indicators in defining a research problem?
Does the question in your mind have no answer yet?
Do the articles online not clearly specify solutions?
Are the solutions not yet tested in the local setting? Or answers to the problems still factually contradictory? Are there any incidents which require explanation?
Indicators of a good research problems. Are you interested with the problem? Can it benefit a group of people? Is it new and fresh? Can it be completed in a defined period of time?
Can develop guide questions? Can it improve a system or program? Are you really determined to finish the study?
Think smart. Like, share, and subscribe.
All right. Well, interesting topic here. There's 108,410,579. Now, that's the estimated population of the Philippines as of the latest. Now, what are the challenges to population growth? How are we coping with a growing nation? What are the programs in place to help cope with population growth? You mentioned like this week that you're doing all of this
Yes.
information in terms of the family planning. What are the other programs?
Well, we work number one on responsible parenthood and family planning. It's our support to the Department of Health in terms of the implementation of the RPRH law. Over the next 4 years, we hope to bring on board 4 million women. That's a big task for us, and the president has given us really a tall order to do that, and we're working hard on that. The second program that we have is on adolescent health and development. And the major issue for us today is the growing number of minors and adolescents actually who give birth every year. It's remained at over 500 for the last 8 years. And this is growth that we've been monitoring ever since we started implementing the RPRH law, and noted that it has really not gone down significantly.
Hello students, this is Mel and Tim. Today, our lesson is all about cell membrane and cell transport. All cells have cell membranes that serve as flexible barrier. Function: maintain homeostasis within the cell. Cell membrane or plasma membrane, composed of 2 layers of phospholipids with embedded proteins. With hydrophilic head or water-loving and hydrophobic tail that repels water. Fluid mosaic model explains the structure of the cell membrane. Cell membrane structure composed of glycoprotein, carbohydrate, glycolipid, cholesterol, integral protein, peripheral protein, channel protein, alpha helix protein, and globular protein. Cell membrane function is gatekeeper that allows certain materials in and out of the cell. The movement of materials in and out of cells or cell transport can be passive or active. Passive transport does not require energy, and materials move from high to low concentration. Example is osmosis. Water will travel from high concentration to low concentration. Osmosis, the diffusion of water across the membrane. Water will move in and out of cells and manifest in various solutions.
Hypertonic and a hypotonic solution. Take a look at these red blood cells and the effect of different solutions. Let's figure out why sometimes a red blood cell stays the same, sometimes it shrinks, and other times it swells. To begin with, it'll be helpful to know the difference between a solvent and a solute. Let's look at coffee. The solvent is what the solute is dissolved into. The water is the solvent, and the cream, sugar, and coffee are the solutes. Now, in our example, we're going to use salt water. In the example, the blue circles is the solvent, water, and the white circles are the solute, salt. An isotonic solution is equal. Iso meaning equal, and tonic meaning strength. So the solute and the solvent will travel in and out of the cell equally, and the cell will stay the same. It's equal. Now, a hypertonic solution is different. Hyper meaning above, and tonic meaning strength. And notice that there are more salt or solute than solvent outside the cell. As a result, you have a higher concentration of solvent inside the cell. So the solvent travels out of the cell and the cell will shrink. The solvent always travels from high concentration to low. Now, hypotonic, hypo meaning below, tonic meaning strength, and notice there are very little solute outside the cell and a higher concentration of solvent outside the cell. So the solvent flows into the cell, remember high to low, and the cell will expand. I hope that's been helpful in knowing the difference between isotonic, hypertonic, and hypotonic.
Hello grade 11 students of SLSPI. This is Ma'am Christine. If you are in trouble on how to make your research title, today, let's have an overview on how to conceptualize a research problem. College research versus classroom experiment. Please take a look. Precaution. First sound like a good research problem. However, 1/nth probability that someone in this globe has them met. Thinking of research topic. Where do you find your research problems? In your brain, use your logic reasoning. In your home. In your community. Rethinking the research topic. Is T, O, E. Stop redundancy. Example. (2017) Roots as dishwashing liquid. 2018 These as dishwashing liquid. 2019 Flowers as dishwashing liquid. Stay connected for our next topic on how to write a research title. The health of population is dependent to population size. To increase population size, we need birth and immigration and decreases through deaths and emigration. There are also factors affecting population growth. We have the density-dependent factor that limits the growth based on the density of population. Density-independent that relates to chance, example flood or fire, that limits the size of population. In population ecology, we are studying these factors, and scientists come up with models that helps to describe what is happening in a population. Example model in population ecology is the exponential growth model and the looking growth rate. Why population ecology is important? Population ecology provides modeling tools that can be used for predicting community structure and dynamics. Population ecology is the study of these and other questions about what factors affect population and how and why a population changes over time. Population ecology is the study of populations in relation to environment, including environmental influences on density and distribution, age structure, and population size. Characteristics of population. Population size, usually denoted, is the number of individual organisms in a population. Factors that increase population size. Birth and immigration increases population size, while death and emigration decreases population size. Population density. Density and dispersion. Density is measured in two methods: total count method and sampling method. The dispersion of a population is the pattern of spacing among individuals within the geographic boundaries. Like, share, and subscribe to my LinkedIn channel. Thank you. Have you ever wondered what we are all made of? All life is made of cells. They come in thousands of shapes and sizes. The human body has over 100 trillion of them, carrying out the dance of life. They are so tiny that 10,000 could fit on the head of a pin. The cells in our body undergo over 500 quadrillion chemical reactions every second. Their amazing biochemistry has nurtured Earth for billions of years and protected us from harm. They ask only that we take care of ourselves. Our science has barely begun to understand them. Let's explore cells.
Hello students, this is our second lesson and this is all about the cell, the fundamental unit of life. So to start with, what is cell? Now cell is the basic structural and functional unit of living organisms. In other words, cell make up living things and carry out activities that keep a living thing alive. Based on the cell theory, so when we say cell theory, it's a collection of ideas and conclusions from many different scientists over time that describe cells and how cells operate. Number one, all known living things are made up of one or more cells. All living cells arise from pre-existing cells by division. The cell is the basic unit of structure and function in all living organisms. We have here cell theory timeline. 1665, Robert Hooke discovered cell while looking at a thin slice of cork. In 1674, Anton van Leeuwenhoek laid the foundation of plant anatomy and became an expert on animal reproduction. He discovered blood cells and microscopic nematodes and studied the structure of wood and crystals. He also made microscope to view specific objects. In 1883, Robert Brown, he is a Scottish botanist best known for his descriptions of cell nuclei and of the continuous motion of the minute particles in solution, which came to be called the Brownian motion. In 1835, we have Felix Duhardin made his largest contribution to the cell theory when he proposed the classification of a new type of single-celled organism which he called rhizopoda, which are now known as protozoans. In 1838, we have Matthias Claden proposed all plants are made up of cells. And in 1839, we have J.E. Purkinje named fluid content of the cell protoplasm. Now, in 1839, Theodore Swan proposed all animals are made up of cells. In 1845, we have Karl Heinrich Braun revised the cell theory with his interpretation that cells are the basic unit of life. In 1855, we have Rudolf Virchow proposed all cells arise from pre-existing cells. There are organisms that are made up of only one cell, and these organisms are called unicellular. We have here an example, a euglena, a paramecium, and yeast. An organism that is made up of more than one cell is also called multicellular organism. Examples are plants, animals, and fungus. So this is the picture of an multicellular organism under a microscope. Okay, the picture of the leaf cell and a muscle cell. Now, cell size vary in terms of most cells are very small, microscopic, and some may be large or macroscopic. Now, the unit used to measure the size of the cell is called micrometer. So when we say micrometer, 1 micrometer is about 1/1000 millimeters. Now, the smallest cell, we have the mycoplasma, which is about the size of 0.1 micrometer. So mycoplasma is a genus of bacteria that lack a cell wall around their cell membranes. And we have also the largest cell, which is in the form of ostrich egg. So the size is about 18 centimeters. Now, female sex cells are just as complicated. Now, the largest egg produced by a living animal belongs to the ostrich. It is 20 times heavier than a chicken's egg and measures roughly about 15 centimeters tall and 13 centimeters wide. So this is the size of the cell in humans. So the smallest cell, the sperm cell, about 5 micrometers. The largest cell, which is about 120 micrometer in a form of the ovum or the egg cell. And we have the longest cell, which is the nerve cell, that is about 1 meter. So cell also vary in shape. Now variation depends mainly upon the function of cells. Some cells like euglena and amoeba can change their shape, but most cells have a fixed shape. So human red blood cells, example we have here, are circular biconcave for easy passage through human capillaries. This one is the nerve cell, or the nerve cells are branched to conduct impulses from one point to another. Now the other one, the human WBC or the white blood cells, can change their shape to engulf the microorganisms that enter the body. So the detailed structure of cell has been studied under compound microscope and electron microscope. Now certain structures can be seen under an electron microscope, and the structure of a cell as seen under an electron microscope is called the ultrastructure. Now, the compound microscope can actually magnify up to 2,000 times, while the electron microscope magnification is about 500,000 times. So, the different parts of the animal cell. So, let's have an overview. Here's the nucleus. Another one is we have the Golgi body. We have here the vesicle. This one is the plasma membrane. We have the mitochondria, we have the cytoskeleton, we have the centriole, we have the lysosome, we have the cytoplasm, the rough ER, the smooth ER, and we have the nucleolus. For the plant cell, we have, of course, the nucleus, we have the Golgi body, we have also the vesicle, we have the lysosome, the plasma membrane, the mitochondria, we have the chloroplast, we have the cell wall, the vacuole, the smooth ER and the rough ER, and together with the nucleolus. For the bacterial cell, we have the capsule, okay, we have the cell wall, we have the plasma membrane, we have the cytoplasm, we have the flagellum, we have the food granule, the plasmid, which contains the DNA, we have the ribosome, the nucleoid, and the pili. So if we study a cell under a microscope, we would come across three features in almost every cell. And these three features are the plasma membrane, the nucleus, and the cytoplasm. So all activities inside the cell and interactions of the cell with its environment are possible due to these structures. So let's start with the plasma membrane. So the plasma membrane is extremely delicate, thin, elastic, living, and semi-permeable membrane. So made up of two layers of lipid molecules in which protein molecules are floating. So in terms of thickness, this varies from 75 to 110 angstrom. Okay? This is angstrom. So angstrom is about 1 times 10 to the negative 10 meters. So functions, first, maintains a shape and size of the cell, protects internal content of the cell, regulates entry and exit of substances in and out of the cell, then maintains homeostasis. For the cell wall, this one is a non-living and outermost covering of a cell. So it is also found in the plant and the bacterial cell. Can be tough, rigid, and sometimes flexible. So made up of cellulose, hemicellulose, and pectin. So maybe thin or thick, multi-layered structure, and the thickness varies from 50 to 1,000 angstrom. In terms of its function, this provides definite shape, strength, and rigidity, prevents drying up of cells, helps in controlling cell expansion, protects cell from external pathogens. The nucleus. So the nucleus is a dense spherical body located near the center of the cell. The diameter varies about 10 to 25 micrometers, present in all cells except in the red blood cell and the Schiff tube cells. So the red blood cells or the RBC lacks nucleus. It's have a well-developed in plant and animal cells, underdeveloped in bacteria and the blue-green algae, and most of the cells are uninucleated, meaning having only one nucleus. So few types of cells have more than one nucleus. The best example, we have the skeletal muscle cells. So the nucleus has a doubled layer covering called the nuclear membrane. Okay, the nuclear membrane, and the nuclear membrane has pores of diameter, okay, pores of diameter about 80 to 100 nanometers. So colorless densa present inside the nucleus known as the nucleoplasm. The nucleoplasm contains round-shaped nucleus and network of chromatin fiber. So fibers are composed of the DNA or the deoxyribonucleic acid and the protein histone. So these fibers condense to form chromosomes during the cell division. Now the chromosomes in the nucleus, so the nucleus contains the chromosome. Now the chromosome contains the stretches of the DNA, and these are called genes. Now, genes transfer the hereditary information from one generation to the next. So the function of the nucleus is to control all the cell activities like metabolism, protein synthesis, growth, and cell division. So nucleolus synthesizes RNA to constitute ribosomes and also store hereditary information in genes. Next one is the cytoplasm. So the cytoplasm is the jelly-like material formed by 80% of water. Present between the plasma membrane and the nucleus, contains a clear liquid portion called the cytosol and various particles. So the cytosol is the liquid found inside of cells, and it is the water-based solution in which organelles, proteins, and other cell structures float. So the cytosol of any cell is a complex solution whose properties allow the function of life to take place. So particles are proteins, carbohydrates, nucleic acid, lipids, and inorganic ions. So also contains many organelles with distinct structure and function. And some of these organelles are visible only under an electron microscope. Now, granular in dense in cells, animal cells, and thin in plant cells. So the endoplasmic reticulum. So we have two types, the rough ER and the smooth ER. Now, in ER or the endoplasmic reticulum, okay, is the network of tubular and vesicular protein or vesicular structures which are interconnected with one another. Now, some parts are connected to the nuclear membrane while others are connected to the cell membrane. So two types, we have the smooth and the rough. Smooth in a sense that lacks ribosome, and rough because of studded with ribosomes. Now, function of ER gives internal support to the cytoplasm. Rough ER synthesize secretory proteins and membrane proteins. The smooth ER synthesize the lipids for cell membrane. Now, in liver cells, the smooth ER detoxify drugs and poisons. In muscle cells, the smooth ER store calcium ions. For the Golgi body, so the Golgi body discovered by Camillo Golgi formed by stack of 5 to 8 membranous sacs. Now, sacs are usually flattened and are called the cisternae. Has two ends. We have the cis phase situated near the ER or the endoplasmic reticulum and the trans phase situated near the cell membrane. Functions: modifies, sorts, and packs materials synthesized in the cell. Delivers synthesized materials to various targets inside the cell and outside the cell. Produce vacuoles and secretory vesicles, then form plasma membrane and lysosome. So how Golgi bodies work? Now the Golgi bodies receive proteins and lipids from the rough ER. Okay, from the rough ER. Now it modifies some of them and sorts, then concentrates and packs into sealed droplets called the vesicles. Now this one, the vesicles, if they're ready for transport to their next destination. The next one is the lysosome. Small spherical single membrane sac found throughout the cell or the cytoplasm filled with hydrolytic enzyme, occur in most animal cell and few in type of plant cells. Now, for the function of the lysosome, first, help in digesting of large molecules. Protect cell by destroying foreign invaders like bacteria and viruses. Degradation of the worn out organelles, then in dead cells perform autolysis. The next one, we have the vacuoles. Now, vacuoles, we have single membrane sac filled with liquid or sop, okay, which contains the water, the sugar, and ions. In animal cells, vacuoles are temporarily small in size and few in numbers, while in plant cells, vacuoles are large and more in numbers. Okay, may be contractile or non-contractile. Now, functions: store various substances including waste products, maintain osmotic pressure of the cell, store food particles in amoeba cells, and provide turgidity and rigidity to plant cells. For the mitochondria or the mitochondrion, the singular form of the mitochondria is a small rod-shaped organelles bounded by two membranes. We have the inner and we have the outer membranes. Now, the outer membrane is smooth and encloses the contents of the mitochondria. The inner membrane is folded in the form of shelf-like inward projections called the cristae. Inner cavity is filled with matrix, which contains many enzymes. Contain their own DNA, which are responsible for many enzymatic actions. Now, the function of the mitochondria is to synthesize energy-rich compound known as ATP, where the ATP stands for adenosine triphosphate. Now, these ATP molecules provide energy for the vital activities of living cells. Plastids. Now, plastids are double membrane bound organelles found inside plants in some algae. So they are responsible for activities related to making and storing food. They often contain different types of pigments that can change the color of the cell. Now, plastids can be chromoplast, chloroplast, or the leucoplast. So for the chromoplast, these are plastids, heterogeneous organelles, responsible for the pigment synthesis and strange in specific photosynthetic eukaryotes. So the chloroplast is responsible for the photosynthesis, which contains the photosynthetic pigment called the chlorophyll, while the leucoplast are plastids that are considered to be colorless. So this includes the three types. Okay. So for the chromoplast are plastids that produce and store pigment. So they are responsible for different colors found in leaves, fruits, flowers, and vegetables. Example, carrot contain the pigment carotene. The mango contains the pigment xanthophyll, and the tomato contains the pigment lycopene. For the leucoplasts are colorless plastids that store food. So they are found in storage organs such as fruits, tubers, and seeds. Example, we have here the potato tubers that contains the food starch. So the food is in a form of starch. The maize grains, we have the food is in the form of protein. And for the castor seeds, the food is in the form of oil. Now, the chloroplast is a double membrane-bound organelles found mainly in plant animals, in plant cells, I'm sorry. Usually spherical or discoidal in shape, shows two distinct regions. We have the grana and the stroma. So granum is the singular form and we have the stroma. So the grana are stacks of the thylakoids. Okay, this one stack here is known as the thylakoid. So that set or stacks of the thylakoid is called the grana. Okay, thylakoids contain the chlorophyll molecules which are responsible for photosynthesis, and the stroma is the colorless dense fluid. So, in the case of the photosynthesis, the light-dependent reaction happened in the thylakoid part of the chloroplast, while the light-independent or the dark reaction happened in the stroma part of the chloroplast. Now, the function is to convert light energy into chemical energy in the form of food. That is from radiant to chemical, provide green color to leaves, stems, and vegetables. Next is we have the centrosome. So the centrosome is the membrane-bound organelle present near the nucleus. Consists of two structures called the centrioles, and centrioles are hollow cylindrical structures made of microtubules. Now, centrioles are arranged at right angles to each other. So, function is to form spindle fibers which help in the movement of the chromosomes during cell division. So, it also helps in the formation of the cilia and flagella. So, the cytoskeleton formed by microtubules and microfilaments, and microtubules are hollow tubules made up of protein called the tubulin. So microfilaments are rod-shaped thin filaments made up of protein called actin. Now functions determine the shape of the cell, give structural strength to the cell, and responsible for cellular movements. So the differences between the prokaryotic cell and the eukaryotic cell. Now, when we say prokaryotic cell, we're talking to a cell that is considered to be unicellular, while the eukaryotic one is multicellular cell or many-celled organism. Now, for the case of the prokaryotic, first we have there, nucleus is underdeveloped, while in the eukaryotic, nucleus is well developed. Next is only one chromosome is present in the prokaryotic while more than one chromosomes are present in eukaryotic. Membrane-bound organelles are absent. Size ranges from 0.5 to 5 micrometers. And examples of prokaryotic cells, we have bacteria and blue-green algae. So while in the eukaryotic cell, membrane-bound organelles are present, size ranges from 5 to 100 micrometers, and examples are all other organisms except bacteria and the blue-green algae. So the animal cell comparison between the animal cell and the plant cell in terms of size, the animal cell is smaller than the plant cell. Cell wall is absent in the plant cell, in the animal cell, but cell wall is present in the plant cell. Now, plastids are absent in animal cell, while plastids are present in the plant cell. So, vacuoles are smaller in size and less in number in animal, while plant vacuoles are larger in size because this one is for storage of food. Centrioles are present in animal, while centrioles are absent in plant cell. Okay, I hope you learned something new today, and don't forget to take down notes and get ready for our next discussion. Okay, that's all. Hello students, this is our first lesson in environmental science entitled An Introduction to Environmental Science. I hope you will watch the video and do some note takings. This lecture will help you understand first the meaning of the term environment, the importance of natural resources, that environmental science is interdisciplinary, the scientific method and how science operates, some pressures facing the global environment, and sustainability and sustainable development. All the things around us with which we interact includes the living things like animals, plants, forests, fungi, etc. The non-living things like for instance the continents, oceans, clouds, soil, and rocks. Our own built environment, this includes the buildings, human-created living centers, and the social relationships and institutions. We humans exist within the environment and are part of nature. Our survival depends on a healthy, functioning planet. The fundamental insight of environmental science is that we are part of the natural world. Our interactions with each other matter a great deal. Humans depend completely on the environment for survival. Enriched and longer lives, increased wealth, health, mobility, leisure, and time. But natural systems have been degraded. Pollution, erosion, and species extinction. Environmental changes threaten long-term health and survival. This is environmental science. This is the study of how the natural world works, how the environment affects humans and vice versa. And with environmental problems come opportunities for solutions. Natural resources are vital to human survival. When we say natural resources, these are substances and energy sources needed for survival. We have two categories, the renewable and the non-renewable one. Renewable resources perpetually available, like for instance sunlight, wind, wave energy, renew themselves over short periods of time, like timber, water, and soil. Take note, renewable resources are resources that can be destroyed. The non-renewable resources are the resources that can be depleted easily. For instance, we have the oil, the coal, and the minerals. The world population is increasing, more than 6.7 billion humans. And why so many humans? Thomas Malthus said population growth must be controlled or it will outstrip food production. This also may lead to starvation, war, disease. Population growth has disastrous effects that is based on neo-Malthusians. Human survival depends on how we interact with our environment. Our impacts are now global. Many great civilizations have fallen after depleting their resources. Look at the picture, the lesson of Easter Island. People annihilated their culture by destroying their environment. Now the question is, can we act more wisely to conserve our resources? Environmental science also talk about how the natural world works. Environment, its impact to humans, and humans, its impact to the environment. Its goal is to develop solutions to environmental problems. Environmental science is an interdisciplinary field. It includes the natural sciences, and this give an information about the natural world, specifically the environmental science programs. For the social sciences, they study human interactions and behavior. Environmental studies programs. Now let's talk about the nature of science. Now, science is defined as a systematic process for learning about the world and testing our understanding of it. This is also defined as the accumulated body of knowledge that results from a dynamic process of observation, testing, and discovery. Science is essential specifically to sort fact from fiction, develop solutions to problems we face. Now, there are policy decisions and management practices. How about this one, the application of science? When we say application of science, we're talking to technology. Now, scientists examine how the world works by observing, measuring, and testing. This involves critical thinking and skepticism. Observational or descriptive science. This talks about when scientists gather information about something not well known or that cannot be manipulated in experiments. Astronomy, paleontology, taxonomy, molecular biology. Hypothesis-driven science. Research that proceeds in a structured manner using experiments to test hypotheses through the scientific method. Now, when we say scientific method, here are the steps. It started with the observations, followed by questions, hypotheses, predictions, testings, and results. Now, when we say a technique for testing ideas, a scientist makes an observation and asks questions of some phenomenon. The scientist formulates a hypothesis, and when we say a hypothesis, this is actually a tentative solution to a problem, a statement that attempts to answer the question. And this is used to generate predictions. And the results will either support or reject the hypothesis. Now, in testing the predictions, we need to conduct an experiment. When we say an experiment, it is an activity that tests the validity of the hypothesis. We need to consider variables. We have two variables involved. We have the independent variable and the dependent variable. When we say independent variable, a condition that is manipulated or a variable that can stand alone. The dependent variable is the variable that is being measured. We have the term controlled experiment. So when we say controlled experiment, one in which all variables are controlled or constant. So this is what we call the unmanipulated point of comparison, while treatment is the manipulated point of comparison. Now, these experiments may lead